And now I'm going to talk about perhaps the most innocent way in which results can be wrong, which is just a straightforward stuff up. Scientists are human, humans make mistakes, scientists therefore make mistakes. And how big of a problem is this? There have been some very big examples. Perhaps the most influential one was Reinhardt and Rogoff's economic paper, Growth in a Time of Debt. This is written just after the global financial crisis in 2008. And what they showed is that countries which had a lot of debt had very slow economic growth after this. This is incredibly influential, and it led, in fact, to many governments, or at least it contributed to many governments deciding to do savage spending cuts or austerity to try and cut their debt level to help them grow faster. The only problem was their entire result was caused by an Excel spreadsheet error. They had accidentally not included all the countries in their spreadsheet, and if they included all the countries they were supposed to be analysing, this is purely a typo in their spreadsheet, then the result went away. Whoops! I mean, you can all see how this happens, but how common are these things? Well, there are ways to look for it. One technique is, it's actually a software called StatCheck. Often when you're doing statistical analysis, there's going to be a whole bunch of numbers. There might be, we find an R value of this and a P value of that, and whatever it might be. And so you can check that these numbers are all consistent. If you've got this R value and that sample size, what P value should you get? And they've done a survey of over 30,000 psychology papers and discovered that over half of them had stats errors of some description, that different stats were inconsistent. You should have been able to derive this from that, and in fact you don't. And most of the time these were very minor errors and didn't affect the major result, but in 13% of these the error was so major it actually completely changed the result of the paper. Whoops! It's a lot of stats errors. Another way to check these things is what's called granularity. If, for example, you ask 20 people to rate something on a scale of 0 to 1, then the average must be a multiple of 0.05. Just having 20 integers in that range and averaging them, dividing by 20, it's going to give you something that's 0.0, 0.05, 0.1, but not 0.008 or something like that. Again, a whole bunch of psychology papers are put through this analysis, and it was discovered that 50% of them fail this test at least once, that there's some average that can't possibly have been obtained from the actual data. And it could be fraud, but it's probably more likely just to be a simple stuff up. They did something wrong in their computer code or their spreadsheet, or just added up numbers wrongly. This actually included the original paper on cognitive dissonance, which is one of the most influential papers of all time. That turns out to have these sort of errors. There's something wrong on the data, though, as it was taken back decades ago. It's very hard to find out what it was now. Another example of a stuff up is in biology. A lot of biologists use cell lines, particularly in cancer research. Someone might isolate some cells, human cells or animal cells that have a particular cancer, and then they'd let them reproduce, and these can then be used to do experiments on, to analyse ways to deal with the cancer. And these are almost immortal cell lines. The first were created back in the 1950s, and some of them have been propagated ever since. So one lab might give some of these cells to another lab, and they will then allow them to reproduce in an appropriate medium and keep using them over years, and then maybe pass them on to other labs. So there's these long lineages of these cell lines, which are a basic resource, particularly for lots of cancer research. The trouble is, it's easy for these things to go wrong. It could be simple mislabeling. Someone puts a wrong label on one of these things, and then for decades afterwards people might be using the wrong cell to do their research. They might be using a monkey tumour rather than a human, or the wrong sort of cancer, ovarian cancer rather than breast cancer, or whatever it might be. Or it can be more subtle than that. You might have in the same lab different experiments on different forms of cancer, and maybe someone uses the wrong pipette and puts it in the wrong culture, and that means a few of the cells from one line get introduced to the other one. Might not be a problem, but these cells reproduce at different rates. If you put some cells from something that reproduces faster, even a few of them before long, they'll take over your entire mixture, and your cell line that you might think was monkey cells exposed to a particular cancer might actually be human cells with quite different cancer, and might have been for decades. This has been known by the biologists for a long time, but it still keeps on happening. Tens of thousands of papers with hundreds of thousands of references have been traced back to contaminated or wrongly labelled cell lines, and therefore are complete crap. And people keep on citing them because they often don't go back and check whether it was correct or not. In some studies, over 50% of cell lines used in China were found to have been contaminated in some way, shape or form. So it's basically because people are not being careful enough and are not going through the very difficult task of testing. They don't test before every time they use one of these cell lines whether it's the correct cell line. Basically, a whole field of research may be complete garbage. Then, of course, there's software. A really good software developer has between 1 and 15 bugs per 1,000 lines of delivered code. Most of the people writing scientific software are not really good developers. They're often graduate students who've just taught themselves to program, and so it's much worse than this. So there is absolutely no doubt whatsoever that the scientific code used to do all sorts of scientific things, I mean, many big projects from the Large Hadron Collider to supercomputer simulations of bomb explosions, they're all using global warming simulations. These are all millions of lines of code, and there will be bugs in them. No one really knows how many scientific results are affected by this. Now, actual computer code is better than Excel spreadsheets because it has all sorts of debugging tools, whereas Excel spreadsheets are much worse. There's lots and lots of bugs like the Reinhardt and Rogoff bug in Excel spreadsheets. You've got to test these things, and you've got to ask how rigorous is your testing. In most software, they will try and test it on situations where they know. For example, global warming, global climate models, they'll test whether they can reproduce the effect of the Pinatubo volcano and whether they can model the end of the last ice age. So you've got to think that your software will have bugs, and how do you know those bugs aren't fatal? And that goes down to rigorous testing, but it may not even be possible to test it that rigorously. And part of the trouble is there are actually incentives to not be careful about these things. People have looked at when there have been results due to cell lines or statistic stuff-ups, and nearly always they tend to err on the side of giving people what they were expecting to get. So maybe if you get what you expect to get, or an exciting result, you don't bother checking too hard, but if you're not getting what you expected or it's not very interesting, then maybe you check harder. If you're being sloppy, you're going to get results. A scatter big or scatter small, and some fraction will scatter big and therefore give you an exciting result that you can publish and get a prestigious journal article. So in some sense, if you're a lab manager, you want your lab to be sloppy because that means you're going to get more exciting results. If you're very, very careful, all your results are probably going to be boring because most real results are boring. So being ultra careful takes lots of time and money and effort and doesn't necessarily make your results look more exciting. So there's a big incentive to be sloppy. Now, I'm an astrophysicist, and I know that in Australian astrophysics there is a very strong culture of you've got to be careful. There's often the nudge and the wink, that person's a bit sloppy. So the counterbalance to all this is that you can get a reputation for doing slipshod, not very careful work, and that can be fatal, and I've seen it be fatal to people's careers. You've got to have a good reputation for being careful. So you always think, okay, what could have gone wrong here? Could have been this, could have been that, check this, check that. It's slow, methodical, tedious, but doing this is very much the culture when you're at least in the fields where I know, in physics and astronomy in Australia. Absolutely, this is a culture. You won't get a PhD if you're careless about these things. It's part of the supervisor's job to inculcate you in this very careful approach. But metrics like publication numbers, citations, media mentions reward careless science, and that brings in the money. And so there is a lot of pressure, and whole fields have succumbed to it. So it's a battle between the careful conservative culture of check, 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 make absolutely sure your results, and all the incentives which reward the quick, flashy, important result. So it's no wonder a lot of people don't be that careful. Quite apart from the solutions. Publish your raw data so people can check things. Most people don't do this. Publish your data analysis code if you've produced a spreadsheet or often these days a Jupyter notebook or something like that to analyze your data. Publish it. Maybe as an appendix so other people can download your code, download your spreadsheet and check your results. Likewise, the referee can actually run your code and check it all. In fact, arguably, all papers should be machine readable, where a computer can go into your paper and actually check it for you. But a lot of people don't want to do this because A, it's more work. They're still wedded to the old style journal, which is entirely in paper, rather than the model style journal, which is mostly digital, but it actually doesn't cost anything to make a huge appendix with gigabytes of raw data. But also it's giving away your edge. Remember that a scientist needs some edge, and that edge might be your software. If you've given it away, anyone else can do this follow-up research faster than you. So there's again an incentive to keep it quiet. There is a final sort of error, which doesn't quite fit into the whole category, which is that let's imagine you did your paper, you did it carefully, you got the right answer, but people might well cite it as saying the reverse. People very often don't have time to read other people's papers very carefully. They just read a summary. And this has caused a lot of problems. COVID-19, for example, we were all told early in the pandemic, keep in Australia 1.5 metres apart. Where did that come from? It came from a misreading of some papers from decades ago. In fact, the disease is mostly spread by particles that can go far more than 1.5 metres. It's probably not a bad thing to keep apart, but there's nothing magic about 1.5 metres. And maybe paying too much attention about keeping people 1.5 metres apart, but they weren't paying attention to more important things like airflow and sterilisation of the air and filtering of the air. The original papers that all this theory were based on actually had the right answer, but people had summarised them, approximated them, and the conventional wisdom, no one ever went back and looked at the original papers. They just thought this is what was going on because they hadn't read it. Another famous example is diet guidelines. For a long time people said the most dangerous thing in your diet is saturated fat. It turns out the most dangerous thing in your diet is sugar. A lot of the research proving that fat was bad was commissioned by the sugar industry. But again, the original papers were nuanced and careful and didn't make mistakes, but people didn't read them very carefully, and people just looked at what other people said about the original papers rather than going back and read it, and so a wrong idea came through. So go back and look at the original papers. They might, if they're correct, they might tell you something interesting. Don't just rely on whatever everybody else tells you. So this is another mistake.